Good morning, sir.
Good morning.
Good morning.
Good morning, Professor Kathiresan. How are you?
Good morning, sir. How are you, Dr. Chinnasamy? You are now principal or you are shifted
Very fine.
to some other place?
No. I'm no more a principal at Vanavara. Now I'm Karunya as a faculty.
Okay.
Dr. Chinnasamy, good morning.
Very good morning, doctor. How are you?
You are traveling?
Fine. Are you still
Fine.
abroad or have you come back?
I'm here only.
You are here only.
I'm in the farm right now in the Karunya. I am in Coimbatore only. Why you went with birds, doctor? What's wrong with you? Good morning, sir.
No,
Sir, good morning.
In Corona,
I was complimenting him.
It's a society program.
I was complimenting him, Chinnasamy. Okay.
Das?
I think Dr. Das, it will be better to start because about 91 people have already
Logged in.
are there.
91.
That's a good number. Yeah.
Should I start now?
Yes.
Yeah.
Respected Dr. Sushil Kumar, President ISWS and Chairman of this webinar session. Respected Dr. J.S. Mishra, Secretary ISWS and Director ICR DWR. Dr. R.M. Kathiresan, Professors and learned speaker of this session. And respected Honorable Dignitary and much dignified with scientist Dr. N.T. Yaduraju, Dr. C. Chinnasamy, Dr. Bhagirath C. Chauhan, and others whosoever are present over here, and others with scientists, colleagues, ladies and gentlemen, and my dear student friend. I, on behalf of the ISWS and ICR DWR, and on my personal behalf, extend a hearty welcome to all of you. Good morning to all of you. We have assembled here to listen to Dr. R.M. Kathiresan, who will speak on a burning topic, alien invasive weeds in India, threat to agriculture, biodiversity and environment. The topic is of paramount importance to weed science. Let us the ball of webinar set rolling. You all know a little background that alien invasive weed in India, it's not new. We have been listening since time immemorial. And there are, as per the record, there are around 173 weed species, invasive species present in India, belonging to 117 genera and 44 families, which constitute around 1% of the total Indian flora. And most of these species, invasive species have originated from tropical America. Around 128 species constitute 74%, and then from tropical Africa, around 11% of these species. And they cause huge loss, economic loss around $1.4 trillion in the world caused by these invasive species. In India, maximum in USA, that is 150 billion US dollar. And in India, it is around 117 billion US dollar. These economic losses are caused by invasive species. With this small introduction, now I invite and request Dr. J.S. Mishra for a formal welcome address to the speaker as well as to the participant. Dr. J.S. Mishra, sir, please.
Thank you. Thank you very much, Dr. Das. Respected dignitaries of the weed scientist, Dr. N.T. Yaduraju Sahab, Dr. Bhagirath Chauhan, Dr. A.S. Rao, Dr. Chinnasamy Sahab, and many more senior colleagues President of the Indian Society of Bead Science, Dr. Sushil Kumar, and Speaker of the Day, renowned bead scientist, Professor of Agronomy, Annamala University, Dr. R.M. Kathiresan sir. As already told by Dr. Das that any alien invasive beads, how they are affecting the agriculture, biodiversity, and environment. Some alien invasive beads have been introduced in India by choice, like this is a water hyacinth as a beautiful flower. But there are many alien invasive beads introduced in India accidentally, like Parthenium, like Felis minor. And they are creating huge loss to the biodiversity and environment. And as already spoken by Dr. Das, that the guest speaker of the day, he has done a wonderful training also, organized training with the FAO on the bead risk analysis, especially for the alien and invasive beads in India. So he is a very renowned scientist, and he has a vast knowledge, especially for the alien invasive beads and bead risk management. So without taking much time, I invite Dr. Kathiresan, but before that, I think Dr. Das will invite the President of Indian Society of Bead Science for further proceedings. Thank you so much.
Thank you, Dr. Mishra Saab, for your very good introductions. And about the speaker also, he told that he is a renowned weed scientist, having a lot of experience in this area. Now I invite and request Dr. Sushil Kumar for his remark, for his presidential remark. Dr. Sushil Kumar, please.
Thank you, Dr. Das. Respected dignitaries, respected all the participants and my colleagues. As you know that this talk is on very important topic, alien invasive weeds. We all are talking about these weeds since long time. And in fact, if you remember about in month of February, we also tried to find out the way to find more significant weeds of the India. And in preliminary understanding or in preliminary survey, what we found that most of the significant weeds are alien invasive weeds. And I think Dr. Kathiresan will throw much light on that, as Dr. Das stated that about 173 weed species have been defined as a alien invasive weeds, particularly on the web portal of biodiversity as well as Botanical Survey of India. So, if you see the list of that 173 alien invasive weeds, you will not alien invasive weeds. Those are 173 are alien invasive plant species. So if you will find the plant species, you will see that most of them belongs to the weeds. More than about 60 to 65% belongs to the weeds. In fact, in the world, there is no plant in the world is you can say unwanted, but this is, we define that whether it will qualify as a weed or whether it will qualify as a plant. So I think that 173 invasive alien plant species are based on the work of Dr. Sandel, and it was published in 2008. So since then, I think there is no update on that list, and Biodiversity Board Authority, they are trying to update that list. And if you will see that list, you will find that there is no mention of Phalaris minor in that one, no mention of Leptochlora chinensis. There are so many weed species which have not been listed in that web portal, means that list of the invasive plant species. Although we are aware that at present, there are many weed species, they have qualified already the status of invasive alien weeds. So with this remark, I think it is better to listen about on that topic by Dr. Kathiresan. And of course, the management of these alien invasive weed species are very important. And you all know that Dr. Kathiresan has done wonderful work on this aspects on many invasive plant weed species. He has individually worked, he has collectively worked with their students. So I think he will be able to justify the topic in great way. Thank you very much. Dr. Das.
Yeah. I don't know what happened.
Actually, somebody has shared a slide.
Yeah. That's
Dr.
why. That's
Sunil
that.
Kumar.
Yeah, that somebody has shared. I don't know who.
Dr. Sunil Kumar, please unshare your slide. Dr. Sunil Kumar.
Yeah. Okay. Now let me introduce. So I am audible.
Yeah.
Yes, sir. Yes.
So let me.
Yes, sir.
Audible. Yeah. Let me introduce the speaker, Dr. R.M. Kathiresan, who is known to all of us, and he is one of us, and a weed scientist of repute. Yet, let me allow to formally say something about his credential and achievement, which will be an eye-opener for the novice weed scientist as well as students. So Dr. Kathiresan acquired his MSc, PhD, postdoctoral research as well as DSc, all specializing in weed science. He did his postdoctorate at Rothamsted Experimental Station, UK as a Commonwealth Fellow and DSc from Annamala University. He has decorated the positions of Head, Department of Agronomy, Dean, and then Director for Center for Research and Development, Annamala University, and he has 35 years of teaching, research, and extension experience. He is consultant for FAO, ED, an expert committee member of National Biodiversity Authority, Ministry of Environment and Forestry. And Dr. Kathiresan has evolved integrated rice, fish, poultry farming technology that has tripled the income of almost 1,600 farmers in Tamil Nadu, and released Sigapi rice variety that could survive recurrent floods in Kerala last year in 1,000-acre area. He is participating in Prime Minister's Skill India program as a technology developer. He handled research project costing around rupees 16 crores, sponsored by World Bank and Bill and Melinda Gates Foundation, USAID, ICR, DBT, and DST, and benefiting around 2,700 farmers in India and even across the border like Nepal. He funded fellowships for students worth rupees 2 crores and added infrastructure for the university worth rupees 3 crore. Dr. Kathiresan has received award from Union Home Minister as well as British Crop Protection Council. He has been extending technologies to SAARC nations, collaborating with NIAES, Japan, FAO, Rome, and ED, Philippines, and Cornell University, USA, and popularizing the cultivation of national flower lotus in rice field as intercrop. So with this brief introduction, now I invite Dr. Kathiresan to make his presentation. Dr. Kathiresan, sir, please.
Thank you. Let me first share my slides, and a very good morning to all the weed scientists, especially Dr. Sushil Kumar, the president of Indian Society of Weed Science, Dr. J.S. Mishra, director DWR ICAR, Dr. N.T. Edraju, Dr. Bhagirat Chauhan, Dr. Prabhakar Shetty, Dr. Chinnasamy, Dr. A.S. Rao, and all the friends known to me very closely in the field of weed science and in agronomy. Thank you very much for this opportunity. And I think that this is a golden opportunity for me because I'm facing my retirement in another couple of months. so that this will be a good opportunity to talk to you all. So my topic, as aligned by the previous speakers, the President of Indian Society of Weed Science and Director DWR, is alien invasive weeds. What happened? Correct. Slideshow. Yeah, alien invasive weeds in India, threat to agriculture, biodiversity, and environment. I don't know whether I will completely justify this topic because this is an ocean, but I've been doing research in certain areas of invasive alien weeds and participating in many forums regarding these invasive alien weeds in India. So let me try to briefly outline the work I have been doing and in common about the alien invasive weeds, the impact of climate change, especially my orientation is towards impact of climate change on these certain invasive alien weeds, not all, because as I said, the invasive alien weeds are proliferating in different areas, different weeds. It's not possible for me to work on all the invasive alien weeds like 173 listed by Dr. Das or by National Biodiversity Authority. I have experience in few. And to start with, the invasive alien weeds are those that have become established where they did not previously occur. And of course, there are ill effects on the environment, the agrobiodiversity, etc. But one thing that is missing in this definition is now we concentrate any plant, whether it is useful or not useful, that is not a question at all. The plant may be useful, but if it is going to be spreading from the area of introduction into other areas, then it has to be rightly designated as invasive. See, let us not bother whether the plant is useful or not. But see, let me again bring back what Dr. Das or Dr. Sushil Kumar said. Actually, if you look into the definition of W.T. Stern for weed, a weed is not so much a botanical as a human psychological category, because a weed is simply a plant which in a particular place at a particular time arouses human dislikes, and attempts are made at its control for the reason that it competes with more desirable plants, or it serves as a host for the pest and disease, or is unpalatable, or is dangerous to domestic bees. So that is the definition given by Stern. It's only a human psychological term, weed. So it is a plant. It could have had some use somewhere, but if it is getting entry into a place where it is not expected, then this word invasive has to be underlined, and we may have to concentrate on that plant. So that is, with that definition, why are we bothered about this like invasive alien weeds? This is because of the Convention on Biological Diversity that makes the states or countries to commit that they will not permit the introduction and spread of any species that is new to a range. And how a plant becomes invasive. It is not that all the plants that enter into a new area or new range becomes invasive, but we say it's Williamson's thumb rule, 1 in 100 will become invasive. So 1 in 100 will become invasive. And how it becomes invasive means it is decided by 3 factors. One is genetic variability, and the other one is climatic and edaphic interaction. The third one is biotic support. So coming to genetic variability, of course, I don't have any example from India, but see, simple understanding is if a plant is genetically invariable, that means it is not ready to mix up with other species. It holds its genetic inheritance, and it doesn't want to mix up with other species and get mingled with the native flora, then it becomes a threat, and its invasive potential is more. But if a plant is ready to mix up with other flora and cross-pollinate with other congeners or other plants around, then the plant becomes less invasive. So lesser the variability of the genetic potential, more is the invasiveness, and more the genetic variability, lesser the invasibility. So there is an example which I have given from North Vietnam. Rubus alsifolius was not invasive, But when it is introduced in Madagascar, it becomes invasive because it was not ready to mingle with the other native flora and it stood by itself and it became invasive. That is with Rubus alcifolius. And again, genetic introgression, sometimes the other way around, introgression with rhododendron catobians in North America, there was no introgression. So rhododendron catobians was an ornamental plant in North America, which was introduced into Europe like Britain, where its introgression with rhododendron pondicum made it more invasive, and rhododendron catobians and rhododendron pondicum, the cross was more invasive, and that is how it became invasive. So this is how the invasive potential is governed by or triggered or influenced by genetic variability. Coming to climatic factors, of course, occurrence of hurricanes can make a plant invasive like Casuarina equisetifolia in California. And likewise, in India, it's a very good example is both water hyacinth and Marsilia quadrifolia. Frequent floods, inundation, and flash floods due to climate change has made this Marsilia quadrifolia and water hyacinth more invasive. And likewise, a lack of physical barriers like rivers. So if there is a river running across, and if the river dries, this invasive alien plant in one region extends into another region. That is in East Africa with Selaginella. Absence of coming to biotic factors, it can be any of these four. Absence of natural enemies makes a plant to become more invasive, or availability of pollinators. Paraprostina is a wasp in America without a pollinator, but when it came to Europe, there was a pollinator for that, and this one became invasive. Allelopathy of companion species. In the native range, the companion species may have allelopathy to arrest the spread of a particular plant, whereas when it is introduced into another new range, if the companion species, they don't have that allelopathic potential to resist the spread, then it may become invasive. And last one is the climatic features, which I have already explained. Invasion of Eichhornia crassipus due to the absence of natural enemies. Say, for example, Eichhornia crassipus was an ornamental plant that was handed over as a complement in New Orleans state in 1876. In New Orleans, the Cotton Expo was organized, and they were giving these water hyacinth plants as a complement to every visitor. By then, they never knew that water hyacinth may become invasive. But later, it was spreading across continents except Europe, and in almost all the continents became invasive. Because in the native range of Brazil, there were so many biological enemies like Neocatenae eichhorni, Neocatenae brucki, Tetranicus stellarius, and other pathogens also. But in the newly introduced range, these natural enemies like Neocatenae eichhorni, Neocatenae brucki, or Tetranicus stellarius were not present. That is how this water hyacinth became invasive. And another one is, say, for example, as a biotic factor, the assimilate partitioning. This is Prosopis juliflora. You know, this plant was introduced telling that this plant cannot withstand rainfall above 1200 millimeter, and this is well suited for only arid regions with less than 500 millimeter of rainfall. Now, this photograph shows this Prosopis juliflora in a lake in Ramnad district of Tamil Nadu, where half of the plant is fully submerged and half is protruding above. What makes this plant to be potential for such alternating climate regimes is this has an assimilate partitioning mode where more assimilates are sent towards the root and less assimilates towards the shoot. So what happens is during the peak summer or when the moisture comes, the shoot dies, but the root remains viable with more of carbohydrate reserves. And in the next season, whether it is absence of flood or when there is optimum temperature, the shoot rejuvenates and props up further with full vigor. Interaction between biotic and climatic factors, replacement of sheep grazing by cattle ranching coupled with frequent above average wet years made Acacia nilotica a problem weed invasive alien species in Australia. Of course, in India, as I said, Eichhornia crassipus is a categorical example. Flash floods with absence of enemies made Eichhornia crassipus an invasive alien species in India to bother about. Trianthema, of course, I have been talking about water hyacinth or Prosopis juliflora, which are only weeds of uncropped areas and social sites and forestry. But whereas coming to agricultural systems, we cannot say that every weed is an invasive alien weed because its origin need to be alien really. So in that context, I have been working on Tranthema portulacastrum, which is an upland weed, which is popular and which is predominating in many of the upland systems throughout India. And this is a weed that is originated in tropical Africa, and it has already invaded Australia, Africa, and Asia. Let us see what makes this Tranthema portulacastrum invasive. Seeds of this weed undergo dormancy during winter and need thermoinduction, and this thermoinduction is by soil temperatures reaching 35 degrees Celsius. Here you see, we have prepared the fields in different months and left it as such without the other plots being disturbed. So one plot will be plowed in January, other plot will be plowed in February, like that, and we have observed the soil temperature and maximum temperature. You can see that the seedling emergence is maximum when the temperature reaches above 35 degrees Celsius. And when the temperature drops down, the seedling emergence is not there. So this is a clear indication that Tranthema portulacastrum is induced by soil temperatures reaching about 35 degrees Celsius. And this is again another clear-cut example of interaction with the climate inducing the invasibility of plants. I got the data of rainfall and evaporation during 10-year segments of 3 10-year segments, 1980 to 1990, 1990 to 2000, and 2000 to 2010. You can look at the annual rainfall that has increased by around 230 millimeters from 1980 to 2000. And 1355 to 1588, almost 230 millimeters increase is there. It is not only increase in rainfall, but it is also decrease in evaporation, which is a very notable point. Say, for example, the evaporation from 2150 has reduced to 1700. So, increase in rainfall by 230 millimeter, decrease in evaporation by around 450 millimeter. So, that means the wet year. Wet year, what do you mean by a wet year? If a year has more than 10% of annual precipitation, it is supposed to be wet. So number of wet years in these segments have increased 5 into 1990 to 2000, 2000 to 2010, whereas it was only 2 in 1980 to 1990. When you say that number of wet years have increased, what does it mean? It means flash floods have gone up. So what has happened? So here is a survey of weeds in rice fields that were fed by or irrigated by 3 different channels during this period from the Lake Viranam. And you can see 1992, what is the status, and 2002, what is the status. Echinoclova clonum was dominating in almost all the rice fields in 3 channels during 1992. Whereas in 2002, it was Leptoclova channel. See, if you want to say that in an agriculture system, a weed has invaded, you cannot simply say that it has invaded by mere observation or mere count. You need to go for phytosociological survey with importance value index or some dominance ratio. Any one of these 2 parameters are important. We have got the survey with the importance value index, and you can see the importance value index of Echinoclova calanum is more in 1992 in almost all the 3 channels rice fields. Whereas in 2002, it was Leptoclova chinensis that is increasing in almost all the rice fields of these 3 channels, and Marsilia quadrifolia also from somewhere from 1 it has gone up to 19 and 20 or 21. The reason is the flash floods and alternating highly submerged condition with dry condition. And this is Marsilia quadrifolia, and this is Leptoclova chinensis, and both increased and decreased runoff could alter floristic composition through invaders. So this is again another categorical example in Tamil Nadu. Eichhornia crassipes have become dominant in areas which have frequent flash floods and submerged conditions, whereas in southern districts where it is exclusively dry, it is Hypomia aquatica under drying habitats. So if it is more dry, the marginal weed Hypomia aquatica becomes dominant. Whereas if it is frequently flooded, then Eichhornia crassipes become dominant. Now coming to Parthenium, Of course, it was observed only in 1955, somewhere around Pune. But in 1975, reports say it is 5 million hectares are observed in India. And in 2010, it has gone up to 35 million hectares. And now what is the status? Only Sushil and DWR can say. Coming to the invasion into agricultural systems from non-crop areas, croplands are observed to be invaded with Parthenium mistrophorus in Andhra, Karnataka, Madhya Pradesh, and Maharashtra. Whereas in orchards, it is observed in apple in Himachal Pradesh, orange in Maharashtra, and mango in Uttar Pradesh. And here also there is an interaction between the climate and the invasive potential of this Parthenium mistrophorus. You can see the importance value percentage of Parthenium mistrophorus in different months and the available soil moisture and mean monthly maximum temperature in those months. And there needs to be a combined factor of available soil moisture above 70 and above 50 and a temperature of around 35. When both of these are combined, the germination and importance value index of Parthenium is more. And you can say there are about 2 flushes somewhere in February, March, and somewhere in September, October it goes. Because only during February and March and September and October, you have the combined factor of available soil moisture of 50% and temperature of around 35 degrees Celsius. Coming to control of Parthenium mistrophorus, again, when we introduce biocontrol agents also, we need to consider so many things. And 2 days back, there was a interaction with the National Biodiversity Authority recent mission on invasive alien plants on different systems. See, initially this agriculturally important insects institute now, which is renamed, earlier it was Directorate of Biological Control. By then the Directorate of Biological Control introduced 3 species, namely Smicronix luteolenthes, Epiblema sternuana, and Zygogramma bicolorata. Smicronix luteolenthes became a pest on sunflower, so it was rejected. So we need to be very careful about host specificity. Unless a biocontrol agent is host specific, we cannot introduce it as a biocontrol agent on a plant, that everybody knows. But the other one, Epiblema sternuana, when it is introduced from Mexico, it could not withstand the temperature and the climate conditions of the local Karnataka or India or Tamil Nadu. So that was also rejected. But Zygogramma bicolorata stood successfully well, and it is a biocontrol agent which has been proved to be very successful, on which Dr. Sushil has much more expertise than me. And cassia species, and again, just 2 days back, there was a reference about this allelopathic plants replacing Parthenium mistrophorus, but we need to be careful. We need to be careful in the sense, I remember Dr. Krishnamurthy from Karnataka, who is solely responsible for introducing this Cassia sericea species for reproduction, replacing Parthenium mistrophorus. He mentioned in a meeting which I was attending some 30 years back, he said, replacing Parthenium with Cassia sericea is like losing one eye to gain the other. So we should ascertain that Cassia sericea will not become invasive on a later date. Until and otherwise, we are sure about that, we cannot introduce a plant to replace Parthenium or anything. Parthenium control with chemicals we have tried, glyphosate, glufosinate ammonium, and these 2 have proved very effective. And I have made an observation, and throughout Tamil Nadu, when we travel towards south, the avenues are planted with tamarind trees, where I couldn't find any Parthenium there. But when I travel towards western districts, the avenues are fully flooded with Parthenium because there is no tamarind tree there. So this tamarind tree should be having some sort of an allelopathic suppression on Parthenium, which we are still working on. So here you see this is a tamarind tree on the avenue where there is no Parthenium there. And mass awareness program we have conducted on Parthenium involving once the Chief Minister of Tamil Nadu made an announcement in the Assembly, Parthenium is a dreaded weed which will be eradicated through Tamil Nadu within 1 year. Then we had that opportunity, we grabbed that opportunity, and we had a mass awareness program involving the state ministers and the district collector, member of the legislative assemblies, which went up very well, and that was published in the Parthenium newsletter published from Queensland. And that was also given a wide publicity, which is the need of the hour. Coming to Eichhornia crassipes, wherein I have been working for the past 30 years, it originated in Brazil. And again, as I said, we have the survey of with phytosociological survey of importance value index from 1990 to 2010. We can see in all the channels of all the 5 channels where we have done the survey, Icomia reptans is only dominating in 1990, whereas it is Eichhornia crassipes in 2010. And this is a lake called Viranam, which is around 16 kilometers wide, and which is a very big lake that has 27 distributaries supporting 18 lakh hectares of rice. Of course, now the water is being diverted for metro water supply to Chennai city. And you can see how water hyacinth has invaded that Viranam lake. And how it is becoming invasive water hyacinth, what are all the invasive traits? Very high seed output and dormant seeds. It has a low herbivore pressure, not that all cattle or anything will feed on that. It has a high phenolic content. Vigorous growth helping to crowd out competing plants and absence of natural enemy. These are the invasive features. Once we study an invasive alien plant, the first and foremost priority is to delineate the invasive features and invasive potentials. Coming to control of water hyacinth through herbicides, because I remember I was once called as a consultant for the Tamil Nadu state government, in which the state agriculture ministry gave a dozen herbicides recommended for water hyacinth biocontrol. But unfortunately, by that time, no herbicide was registered for use on a water body. And I said, you cannot use a herbicide on a water body because it is not registered. But then the Ministry of Agriculture, Environment, Ministry of Water Resources gave us a project to evaluate herbicides for water hyacinth in controlled sites of water bodies. And then we screened different herbicides. And you can see this is untreated control, 2,4-D-sodium salt, imazapyr, glyphosate, paraquat. Up to 35 days we have tried. And almost all the herbicides were effective. There is no difference, much of a difference. Imazapyr was excelling with very quick control, whereas glyphosate and 2,4-D were a bit slow. But all of them were giving good control at the end of 40 days or something. But coming to water quality, what happens to the water quality? All the herbicides were detrimental, especially those which are having very high efficiency. They were very highly detrimental with the minimum or least dissolved oxygen, with imazapyr giving the least dissolved oxygen. But glyphosate was comparatively better in terms of water quality. Of course, glyphosate is now constrained with the ban and all. Among the herbicides tried, imazapyr excelled. However, as regards water quality, imazapyr reduced the water quality, were efficient in different growth stages, did not differ. Whether it is a small plant, say for example, water hyacinth, you can have plants up to 10 centimeter and you can have plants up to 6 feet. So whatever the height or whatever the stature, they don't differ in their response to herbicides, and all herbicides are efficient. Stagnant water body trial, we then switched over our studies to stagnant water body. And this is, of course, I don't want to waste time in giving you the data and all. So this is how we did. We used fish nets to divide compartments in a stagnant water body. And we tried 2,4-D-sodium salt to control how it was looking, 30 days, how it was looking, 50 days. And again, this is with regard to maybe paraquat, effect of percentage of, and what happens to the crops that are irrigated after treating the water bodies with herbicides. Of course, none of them had any detrimental effect on water hyacinth controlled water using herbicides. Evaluation herbicides, okay. They are safe for irrigating to the cropland, that's okay. But what happens if it is an aquaculture, if the water is used for aquaculture or fish culture, then we tried it on fishes. And this is how we tried cement tanks and with the fishes and without fishes, we tried using the herbicides. And these are all the treatments. Fish alone, 2,4-D-sodium salt, with the fish and water hyacinth, glyphosate and paraquat. And you can see fish alone is safe, whereas 2,4-D-sodium salt, glyphosate was safest, whereas all other herbicides had mortality on fishes. Among the herbicides applied, glyphosate has shown least fish mortality percentage. application of 2,4-D sodium salt and paraquat have shown higher fish mortality percentage. And regarding the histopathology of fish organs, almost all herbicides were lethal. 2,4-D sodium salt was causing injury, glyphosate was causing injury, and lesser neurological cells, even glyphosate was not an exemption. So our final conclusion is, if herbicides have to be used for controlling water hyacinth, that water body should not be of use to aquaculture, and it should not be supporting any fish or any other aquaculture organisms. Then we tried, of course, this is my exclusive experiment in Rothamsted. We had a medicinal plant called Coleus zambionicus, and this I have demonstrated, projected or presented in many of the Indian Society of Wheat Science presentations. This is supposed to be having allelopathic suppression, very good. Because say, for example, when we dry the powder of Coleus zambionicus, and when we mix it up in water supporting water hyacinth, you will not believe, if you mix it up by 10 o'clock in the morning, by 3 o'clock in the evening, all water hyacinth plants will be dead and drying out. And I remember my supervisor, John Terry in Rothamsted, said, "You are not doing any research, you are doing some magic." So that is how Coleus zambionicus works. And this is at 25 gram per liter of water, how within 5 hours this water, Coleus zambionicus is drying water hyacinth, you can see. And this is with regard to cut leaf bioassay. Even at 0.1 gram, if it is directly introduced into the leaf, it is active. And then when I came back to India, I started thinking that if these Coleus zambionicus could be directly absorbed by the leaf, that could be some more effect. And we tried combining it with the biocontrol using Neocatina acarnea and Neocatina brucki. And we see this is the powder that we have prepared out of Coleus zambionicus, and we released the insect agent first and then sprayed Coleus zambionicus on that. And this is how it works at 25% foliar spray. Within 3 days, we were able to control water hyacinth. And we tried this in 3 different water bodies, in 12 different water bodies in 4 of the districts. And you can see in January, we released the insect agent. In April, we had the foliar spray of Coleus zambionicus. In July, you can see water hyacinth drying out. And in October 2010, of course, in the next season, there was some rejuvenation, but then we repeated the experiment in January also. Not only that, there is a allelopathy of rice straw on water hyacinth also. So we screened the different rice cultivars for the straw of rice cultivars. And we found that BPT and Punni were very effective in controlling water hyacinth. And this is how the study was taken up with the polystyrene cups. And this is the dry ground powder of straw of different rice cultivars. And this is the data actually, percentage reduction of wheat biomass. You can see based on that, we divided the rice cultivars into highly allelopathic, moderately allelopathic, and less allelopathic. And we proved that on the micro ponds in cement concrete tanks also. Confirmation simulated micro ponds and water up to 0.5 meter depth, chopped straw of rice cultivars BPT and ADT36 were soaked. And this is the result, of course, BPT and ADT36. They had a very high efficacy on reducing the water hyacinth biomass. And then we had an experiment in Sonning-on-Thames in UK with lavender stalks for control of algal blooms. With that, I had the idea of making use of this rice paddy straw into polybags bundled with holes on the polybags. And then we soaked it in the ponds where there were water hyacinth, and we had effective control of water hyacinth. And then another avenue for control of alien invasive plants is attaching a utility tag over the plant. So if somebody finds the plant to be useful, then it will be very easily managed. So we tried it as animal feed, manure, and then we extracted nanofiber. And we mixed the water hyacinth powder into the ration of calves, cattle, and then we found that it never had any ill effect on the blood biochemical parameters of cattle. So we proved that it can be used as a cattle feed, and then it can be used as a compost. And we extracted nanofibers, and we had the data to support that the nanofibers are very effective in making acoustic boards and biocomposed polymers so that it can be used by the industry and water consumption could be cleared by the industry. And next bead, I am coming to honeysuckle mesquite Prosopis juliflora. This is actually a headache and which I wanted to make this opportunity to disseminate the information among the bead scientists here. See, in Tamil Nadu, one politician went to High Court and filed a public interest litigation that Prosopis juliflora is a menace and it should be controlled. Actually, the court gave a very good verdict directing the state government to clear Prosopis juliflora in all the sites wherever they find it. But unfortunately, this forestry department, which has introduced Prosopis juliflora, actually Prosopis juliflora was named as royal tree in 1910. It was imported with the request of Deputy Conservator of Forest, Madras Province, to Queen Elizabeth, and Queen Elizabeth sent this Prosopis juliflora seeds for aerial seeding in dry areas, thinking that it will be a good alternative, giving livelihood options for people in drier areas like Anantapur or Kovilpatti or any other region in Tamil Nadu or Andhra Pradesh. But what has happened from royalty, the Kabi Bioscience Biocontrol newsletter brought out an article recently with the title, The Status of Prosopis juliflora in India from that of royal tree to pariah. And in which actually I had a chapter in FAO on Prosopis juliflora invasion in India, in which I concluded that Prosopis juliflora is like a wildfire, neither deserves an extinction nor a spread without watch. This sentence, this biocontrol news of Kabi has recapitulated. This is a dreadful tree, and these forest people defended that Prosopis juliflora should not be eradicated because it is very useful for people in dry areas, and just because they introduced it, they were defending. And unfortunately, only one agronomist who is from the veterinary university is drafted into the team for giving the recommendation. And he himself, of course, he is my student, and he was not aware of the dire consequences of Prosopis juliflora. And then the High Court partially reversed the verdict, telling that it can be controlled only in controlled environment, and in non-crop areas it can be left as such. So this is how we, the beet scientists and agronomists, are not able to defend our stand fully, and we were not considered equal, and we were not involved in many of the disputes like this. Coming to Prosopis juliflora, it's neotropic in origin. Northern South America, it has originated from Central America and Northern South America. And rate of spread and invasion increased from 371 hectares per annum during 1978 to 92 to 460 hectares per annum. Every year, it has increased from in Sudan. Of course, 230,000 hectares invaded in Sudan now. Whereas in Gujarat in India, the rate of spread is 25 square kilometer per year. Every year it is spreading by 25 square kilometer. And as of now, I have the data only up to 2006, 5.5 million hectares invaded in India. And another defect or another shortfall we have is we don't have any area-wise. This much area is under an invasive alien species, under noted invasive alien species, that we don't have the data. Ecological amplitude, it thrives well. Wide temperature regimes, 10 to 15 degrees Celsius, of course I have mentioned, but now I can say 10 to 45 degrees Celsius in India. Varied rainfall distribution, 150 to 1500 millimeter it can withstand, adapted to wide range of soil types. These are all the adaptations. And see, 63,000 to 9,80,000 seeds per tree, the potential, and 367 kgs of pot per year. And of course, this tree is unique. The calorific value is the highest in Prosopis juliflora among all the trees when it is used as a fuel wood. I don't deny. But at the same time, the pots have very high sugar content, and it is succulent, and it is preferred by the cattle and small ruminants. Temperature, and this is actually the study we have done. When there is a increase in temperature by 2 degrees from 28 to 30 and 30 to 32, you can see the increase in shoot biomass is not that much. It increases from 42 to 47 and 47 to 56 kg only, within some 10 to 15 kg. But look at the root biomass, 1.9, from 1.9, it doubles almost to 4.4. And from 4.4, it triples to 6.2 with 4 degrees Celsius increase. So what it means, The biomass in the root is more, which gives the potential to rejuvenate whatever the climatic extremes on the above ground during the subsequent seasons. Dispersal is aided by animals, intentional introduction and water. And this is the root biomass, which I have been mentioning about. This much of capable biomass is there in the root. The fuel wood availability ranges from 40 kg per tree from 4th year of planting, varies up to 139 kg per tree until 10 years after plantation. My opinion is it may be of very high use, I don't deny that, but it is very highly spreading. It is very highly invasive. And so we need to control that. There is no question of using it, and nobody can contain it within a particular controlled area. It will spread, and for that reason, we need to eradicate Prosopis juliflora. Parts of juliflora have been used as animal feed for cattle, sheep, goats. And of course, I will say, DFID, Department for International Development, Government, United Kingdom, has conducted workshops in India, in Rajasthan, to train people to make use of Prosopis juliflora. That was in early 1990s. And likewise, I am thankful to J.S. Mishra was mentioning that we had a workshop for training the government representatives of Asian countries in weed risk assessment in Annamalai University in 2007. What happened is the trainer gave 2 examples which were very invasive, behaving invasive range extension in Africa, and he tried to score out the potential in India if introduced in India, and our participants scored it out, and it was having very high potential. But unfortunately, after that, we found that both these species were already imported by Central Grassland and Fodder Research Institute, Jhansi. What to do? So we cannot help it. Unless there is a collaborative and joint effort by all the stakeholders, it's very difficult to prevent the introduction and spread of invasive alien species. Problems of Prosopis juliflora. Aggressive invasion of Prosopis juliflora results in suppression of native biodiversity and species richness of habitats such as pastures, woodlands, and arable lands. In water catchment reservoirs, the weed causes increased evapotranspiration loss 9 times. The evapotranspiration loss is 9 times higher. Hinders some primary production sectors. Mesquite pollen is recognized as allergic to human. Higher sugar content of pods causing teeth decay in animals. Presence of Prosopis juliflora along the coast and amidst. See, this is another thing. When there was tsunami affecting or attacking the coast of Tamil Nadu, the fisherwomen who were caught in the floods, they were trying to swim out, but their saris and hair were caught in the bushy thorn of Prosopis juliflora, and they drowned and died. That is the, see, Prosopis juliflora, see, mangroves are sites with very high salt content and water of very high salt content. Even Prosopis juliflora was able to tolerate there and come up very well in amidst mangroves. Control options: mechanical, ploughing or using heavy equipment, slashing and digging out roots, slashing and burning. And we tried 2,4,5-T, glyphosate, trichlopyr, picloram, clopyralid, methods of application, basal bark application, cut stump application, foliar spraying, nothing was effective. And slashing and burning alone was effective compared to slashing and 2,4-D application. Slashing and burning is the only effective thing, and digging out using pocklines or JCB will be more effective. Managing regrowth, and that itself is not sufficient. Once it is dug out using a JCB, we need to rehabilitate the site with the cropping, with prostate growing habitat plants or crops. Biological control agents, there have been so much of biological control agents suggested outside India, but nothing has been tried in India so far. Other invasive alien weeds which I have not worked and which I have not made mention so far is Chromolina odorata, Eupatorium adenoforum, Salvinia molesta, Lantana camera, Mecania macrantha, Mimosa rubicalis. Coming to sleeper weeds. What are these sleeper weeds? Of course, I will say that in the process of invasion, the ecological process of invasion involves 6 different phases. First one is entry. Second one is establishment. Third one is entrenchment. Third one is expansion. Fourth one is entrenchment. Fifth one is explosion. And the last one, sixth one is escape. So a plant would have got introduced, it would have entered, and it would have crossed 3 stages. That is up to expansion. It would have expanded in the introduced area, introduced range, but it would not have exploded. It would not have got into the 4th stage. Such of those plants, such of those weeds are called as sleeper weeds. That means it may become invasive alien weed anytime afterwards. So there needs to be a watch on the sleeper weeds. And in Australia, they are very effective. They spotted 117 sleeper weeds and they were observing it for more than 5 years and narrowed it down to 17 weeds in 2006. And Piptochetum motividensis, they suggested for eradication because it has more potential to become invasive alien, and they eradicated it, of course, successfully. But Hieracium aurantiacum was also a sleeper weed that was spotted for observance, but they observed it and it was not behaving invasive, so they left it. Prediction of weed invasion, traits of a successful invader. It has, see what traits you need to look for if you want to spot it out as an invasive alien plant. High habitat disturbance. It should have very short generation. It should not be leading for a long life. It should have low herbivore pressure. It should have mycorrhizal infections and links, prolific production of easily dispersible seed and vigorous growth. These are all the characters we bring out in the score sheet for any weed risk assessment. And as I said, the process of ecological invasion involves 6 phases: entry, establishment, expansion. These 3 are by the genetic potential and native inherent ability of the plant. Whereas explosion, entrenchment, and escape, it should be aided by the climate or the biotic factor. Aquatic weediness. See, models for predicting invasiveness. This I was making a mention. There had been several models. First one is Hazard model, 1988. That was the first model developed in Australia that considered aquatic or weediness elsewhere, seriousness, poisonous or allergic, vegetative reproduction. These scores I have given. And the next one, of course, Penita model, and then Phelog model. And then, because I'm not concerned very much with weed risk assessment as of now in the topic, I'm just rushing up. USDA model in 2000. But none of these models were adequate to cover exclusively. Then Champion and Clayton model in New Zealand, and they had different scores pattern. And the last one is Chris Parker model, which was considered to be more exclusive, and it considers 4 different ranking scores: A, B, C, and D. A is invasive potential. How do you measure invasive potential? Whether the plant is rhizomatous, climbing, small and readily dispersed seed. If the plant has any one of these characters, it can be called, it can be given full score under invasive potential. Damage potential. If the plant is highly competitive, or if it has an allelopathic effect, or if it has both, then damage potential score can be completed. Geography potential. How far it has extended in other regions. Range extension or ecology is vulnerable, and how far it is going to affect the ranges within the introduced range. That is geography potential. Entry potential, mode of entry, accidental or deliberate. This is considered to be the entry potential, and the score of all these A, B, C, and D, score of the product. Why this is given as a product? Say, for example, it has a high score in invasive potential, okay. It has a high score in damage potential, okay. But it has 0 potential in geography, and it has a high score in entry potential. Then A into B into C, Z is 0, so it becomes 0. We need not bother about it. That is the significance of sum, not the additional factor. It is not plus, it is into. But later on, we found that climate also need to be introduced, and based on Chris Parker's model in 2010, myself and Galbert Becherno developed another model for FAO in 2012. We published it in 2012, which has A, B, C, D, and E. That is, E is climate potential. Invasive traits of a species comprising biological attributes and ecological traits help only up to establishment. Further progress is restricted by native vegetation. Species are favored to get through by nature and frequency of habitat disturbance such as fire, flood, and invasive animals. Of course, I have mentioned about it. Climate change in future would increase the probability for open niches in the ecosystem favoring alien weed invasion. And of course, more appropriate models suggested climatic potential included as E, and that was suggested in 2016, published in 2016. Prevention of invasive weed revolves around the no individual or organization has the right to move an undesirable taxon from where it is to where it is not wanted or to where it will cause environmental and economic harm. And prevention revolves around producing commodities in weed-free area, mitigation of contaminated commodity before shipment, insisting on permits of certificates, legislation and inspection, of course, awareness also. And Parthinim Awareness Week is being very good, very well organized by DWR and Indian Society of Weed Science in India in the 3rd week of August, I suppose. Weed Awareness Week is organized in USA, Weed Buster Week in Australia. And National Entry Early Warning and Rapid Response System for invasive potential is being launched in USA. Climate change in general might increase activity of species currently limited by thermal accumulation at higher latitudes, might spread tropical and subtropical species in temperate areas. Stimulated C4 weeds to a longer extent than C4 crops. Elevated CO2 will stimulate C4 weeds than C4 crops, that is a literature. Relative biomass increase in higher in fast growing than slow growing C3 species. Both in C3, C4, elevated CO2 is going to favor only weeds than crops. C3 weeds and crops in elevated CO2 favored Chenopodium album as against soybean. Elevated CO2 accelerate germination of smaller seeds, and more especially these weeds have smaller seeds. Increase tolerance to temperature extremes, enabling weed dispersal and range extension. Mechanisms imparting tolerance to temperature extremes at elevated CO2 is increased temperature optima for photosynthesis. Of course, temperature optima, whether it is saturation point or this is photosynthesis. C3, there will be higher saturation point and there will be a higher compensation point in photosynthesis with elevated CO2. And reduced stomatal conductance, mitigating the dehydrating effects of temperature stress and exposure to pollutants. Elevated CO2 also favors weedy attributes. Other attributes of climatic change that can trigger invasivity, as I explained earlier, with altered rainfall pattern, that is going to naturally trigger those plants which remain as sleeper weeds or those plants which have very high invasive potential to become more and more invasive. Increasing temperature regimes is also another factor. So this is what I wanted to present to you in chart, and of course I think I have finished within 45 minutes of time as delineated by Dr. Sushil and Dr. Mishra and Dr. Das. Thank you. I will be happy to interact with you if it is required. I thank Indian Society of Weed Science and Directorate of Weed Research, all the weed science colleagues once again for this nice opportunity. Thank you all.
Hello. Thank you, Dr. Kathirasan Saab. This is a very nice and elaborative presentations, and this is really you could share your research derived knowledge as well as experience, and you all agree that the deliberations opened up several dimensions of research and scales of innovations, particularly on the Prosopis juliflora, Eichhornia crassipes, this invasive weed on which he has worked a lot. And he also highlighted the importance of climate change, the model, how the model could be used to characterize the invasiveness of species and so many things. So this is really a useful deliberations. Thank you, Dr. Kathirasan. Now I will invite the suggestions, queries from the learned experts, with scientists, and whosoever present over here. Please interact with the speaker and get clarified. If you have any sort of doubt, query, please come up. Thank you.
Dr. Das?
Dr. Chinnaswamy sir, please.
Yeah, Dr. Das, good morning, and Dr. Kathirasan. Really, it is a good presentation. As usual, you do it extremely well. And I appreciate and congratulate the organizers as well as the presenter, the guest speaker. I have only one apprehension, Doctor. We do qualify this Prosopis juliflora as weed. No doubt about it. It's because of invasiveness. Leave alone the cropped field, probably based on its significance in terms of contributing for the economic importance, as with the basic idea with which the Department of Forestry has introduced from elsewhere. Would you like to contain this one? I mean, based on your own perception, leave alone the cropped field. Even for weeds, we do define that just because the sake of biodiversity or for other concern, other than non-cropped area, except those which are noxious, we are not much bothered and we may not bother about it. So what's your view or opinion about that?
Yes, exactly. I thank Dr. Chinnaswamy for the compliment as well as the comments, and really a very good question, of course. Many of us think that Prosopis juliflora is only a weed of social sites and uncropped areas. And see, the thing is, if Prosopis juliflora comes up initially, say the coverage initially may be around 5 meters, 5 square meters. But it expands its expansion, and within 2 years, the coverage will be around 25 square meters, and it forms thickets in the center. That is the problem with Prosopis. Any problem, any area affected by Prosopis, there will be a strong concentrated thicket in the center, and the periphery only will be accessible for people to move around. They will not be able to move inside the central thicket of Prosopis juliflora. And not only that, if you go into the southern districts like Ramnad or Sivaganga or Koilpatti and such regions, if a farmer leaves his field uncropped, maybe for rain-fed rice for 2 or 3 seasons due to monsoon failure, 3rd year or 4th year, a few plants of Prosopis juliflora comes in that rice field, and 7th year, the field will be completely abandoned by Prosopis juliflora. So incursion into agricultural sites and making them highly untenable for agriculture is quite very frequent phenomenon with Prosopis juliflora. So that is why we are bothered, and I am very much worried about the forestry people safeguarding Prosopis juliflora as if it is a savior.
Yeah, that's fine.
Okay, yeah.
It's a very good explanation you have given. Thank you very much for having permitted me, Dr. Das. And
Dr. Chinnaswamy sir, thank you.
Okay,
So, sir, Dr. Yadav Saab, are you here now?
Well,
Yes, sir.
I
Your
don't have
comment,
any
expert
specific
comments, sir.
questions,
Please, your expert size and comments.
Thank you. I mean, I don't have any specific this thing, and he has, of course, as pointed out by Dr. Chinnaswamy, and many of us will agree that it was a wonderful presentation, very educative, very informative, and mostly drawn from his first-hand experience of working with those weeds. And I think I'm sure he himself admitted that he cannot cover all the invasive weeds. I'm sure that there are more weeds also, as we know that there is a problem of Chromolina odorata throughout India. There is a problem of Bicania micrantha in northeast and in tea plantations, and there is problem of mimosa in Kaziranga National Park. So there are plenty of them. I'm sure that there will be another presentation by somebody to cover those things. And I just want to tell to complete the newcomers and the students is that Dr. Kathiresan, I know, of course, he is a good old friend of mine, and we have been in touch with each other, both academically and personally. And since he's organized an ISWS conference, back in, I don't know, maybe 1980s, late 80s, 2000, okay, 2000. And I have been to his lab a number of times. And I think, we know that attending international conferences is so difficult for us for the simple reason of economics. But I think you should learn from Dr. Kathiresan. He's the one person who has gone to so many international conferences, so widen his own horizon and to learn more things and to network with people. And most of his conferences, if I am right, he has attended at his own expenses. Not many of us, including me, we have not done, and probably many of us won't do. But what I'm telling you, this shows his enormous interest in the discipline and in the pursuit of knowledge. And I think that is one of the sort of takeaways. And another thing is that you all know that he is working in a institution which is not in the mainstream of NARS, National Agricultural Research System, but still working in a non-agricultural university. He established a very good team in his Annamalai Agricultural University, Annamalai University, and he not only made his name, but has really sort of trained so many people, students and young scientists and all. So I think these are the things, he could be don't want to repeat all the contributions he has made that has been already highlighted earlier by Dr. Das and Dr. Mishra and Dr. Sushil Kumar. And I think his work on biological control of, the water hyacinth is well known with using the coleus and all that. And of course, after this thing, I would like to ask Dr. Kathiresan is that such a wonderful, discovery, you can call it, about the active ingredient responsible for such a quick kill of water hyacinth. I've seen myself when I went to his lab, how good it was. And he was under the NEIP project, he was trying to use both coleus extract plus the insects also, so that the penetration of the extract will be more with insect attack. So I would like to ask him, where is it now? Can somebody take it forward about the active ingredient? What are the limitations? So that at least such a good innovation, such a good discovery should not be kept under wraps, kept in the almirah. So that is one thing I would ask him to interact later. Of course, before I wind up, maybe I will not take much time, is that after every webinar, such both ISWS and DWR are organized, I think we leave a lot of homework for Dr. Mishra. I think in every, I'm sure that he is noting it down, and Sushil Kumar also, and wherever possible, I try to address it. Just don't stop it here. I think I can pick it up on the quarantine weeds done by Dr. Moochan, and many, many good things came out. And here also, I would like to say about a few things for the, for the information of Dr. Mishra particularly, is that as pointed out earlier, that there is no data. We have been hearing about these invasive weeds for a number of years, and there is no update on the area coverage, on more importantly is what is the economic and environmental impact. And I have been telling about it, and as a in one of the newsletters when I was the president of Indian Institute of Weed Science, in the presidential message, I left a note about what Australia is doing about working out the economic impact of these weeds. They do it because unless you say that this weed is causing so much of economic damage to the society and to the nation, they will not get grants. But unfortunately, in India, we don't have that problem. We have got government jobs, and whether you research or not, at least your salary is ensured. But that is not good. If you want to contribute, let us sensitize the policymakers about the economic and the environmental impact of these weeds. And so I think I would like Dr. Mishra to take few weeds, not all, few weeds, important ones, and maybe like a collaborative project funded by ICR or somebody, or DST or DBT, can work on this thing. And I think I'll again mention about the CRC weeds in Australia. They got the methodology, they got a publication, which I distributed in that newsletter. I gave the link also for people to download, and you can copy how to do it, because we don't have the expertise. We need to involve other people also, like economists and other people. So that I think it is very important that you can, maybe Dr. Chavan also can comment on that about the CRC publication on how to
Yes.
work out the economic impact of the beads. I think, another thing is the last point, Dr. Das, the last point I want to tell about is that we all know that management of invasive weeds is very, very difficult. And no other method is cost effective and practical. We all know it. Biological control is the only one which is practical because it is economical, it is self-spreading and things like that. But we are not made much headway. So I again taking up on the interaction we had last time is that I would like to ask DWR to collaborate more intensely, more closely with the National Bureau of agriculturally important insect resources. Now they call it NBIR Bangalore, to introduce more weed species and have collaboration and come up with a solution. Otherwise, mechanical and cultural and chemical weed control will not work. Who is going to do all this, for example, to control Mecania in the forest areas, and same Cromolenia in the non-cropped areas? So I think more practical method of management is biological one. I think we need to do that. I think there's a CABI has done some work with using rust fungus for control of Mecania. And I think we need to have more such collaborations with international organizations to bring in expertise, bring in more biological control agents. I think I will stop here, but I'll request Dr. Kathiresan to answer to my question about where his coleus extract stands now. Can somebody, maybe his successors or students can take over? Please comment on that if you don't mind.
Thank you, Dr. Edraju. Thank you for the compliments and all your suggestions on the whole of invasive alien plant species research. Regarding the Coleus umbilicus, actually I was working on isolation of the active principle of Coleus umbilicus in Rothamsted, and we did isolate. And especially in the case of any natural product working on any target organism, maybe an insect or maybe a weed plant, the thing is, it is not a single molecule in most cases that is active on the target organism. It is a combination of conjugated molecules together acting on the organism, and that makes it difficult to isolate the active principle. Of course, we have isolated the active principle, but by that time, the Rothamsted Experimental Station was insisting that we should get into a confidentiality agreement with them. And the reason is, if this active principle is working very effective, 60% of the IPR will come to Annamalai University and 40% to Rothamsted. But at the same time, they were going on for synthetic analogs of the active principle. And if that synthetic analog is more active, 60% will go to Rothamsted and 40% alone will come to Annamalai. But fortunately, and in many cases of natural products, not many synthetic analogs are more effective because of the reason that I said. It is not a single molecule, it is a combination of molecules that is active. That is why I just left that confidentiality agreement, came to Annamalai University and started working on the natural product as a core material using insect agents for biological control. But anyhow, people can still work on that. As I said, not necessarily Coleus umbilicus. There are even rice varieties with their straw, allelopathic on water-hyacinth, and many more plant materials are effective on different plant species. And again, for allelopathy, it is easy to work on water system, aquatic system rather than terrestrial system. Because in the terrestrial system, the soil interface is there. The soil degrades the allelopathic chemical. Whereas in a water system, the allelopathic chemical is directly absorbed by the plant. So I invite my own students, my own successors, and not only that, all other scientists to work on allelopathy in aquatic systems. And this will be a better route.
Thank you, Doctor.
Just 1 second, Dr. Das, if you don't.
Yes,
don't mind. I know one can start, but you have gone to such a high level. I think it must have taken many years of research. So why to waste it? So I still insist that forget about Rothamsted. 1 second. Forget about Rothamsted. there are so many institutes in India, National Chemical Laboratory, Pune, IRA, Agricultural Chemicals Division, IRA. I think still you can rake it up if you don't mind.
Yes, okay.
I think it is
Yeah,
worth
I'll do it.
trying.
Yeah.
Don't lose that advantage, the head start you have. Thank you.
Thank you, sir. Thank you, sir.
Thank
We
you.
are actually, sir, we are, so Dr. Kathirasan, you can have some interaction with Dr. Yaduraju for advancement of this research work further. So now, because we have, I want to pick up some quick question. There is only one question I want to convey, Dr. Kathirasan, that most of the participants, they have conveyed you for your nice, excellent presentation. They have conveyed you best wishes. And one is Dr. Sandeepa Chaudhary. She wanted to know how water has seen threats to species biodiversity. Could you answer?
How water has seen?
How water has seen threat to species biodiversity. So just in quick, you can just pick up this.
Yeah, if water hyacinth is there in an aquatic system, it will obliterate all, say for example, lotus. Lotus is a national flower. It has all medicinal properties throughout its root to stem to its leaf, and it is being used in for different purposes. But water hyacinth has replaced lotus.
Okay. Thank you.
So lotus is an important component we have lost.
Anybody, please, last question, because we are running sort of time. Huh?
Hello.
Yeah, hello.
Yeah, good afternoon, sir.
Good afternoon.
Myself, Dr. Ajay Kumar Singh, working as a principal scientist, AG Bio Company.
Yeah.
We at the R&D center, we are working on some invasive weed for Parthenium, lantana, and water hyacinth. So we have developed some micro herbicide for these three weed. This technology developed in a systematic and very scientific way. We have developed and protected IP for these all three weeds. And we have also got a patent for that all the three technology. That is, we have brought some indigenous species from the water hyacinth. It is showing very host specificity toward the mother plant, and is controlling the weed very nicely.
Okay. Thank you, Dr. for your information. And this is information. It is passed on to everybody. So those who want to pick up research on this bioherbicide, they can have some collaboration with you. You need collaboration also. Huh? You need collaboration.
Sure,
So, okay, sir. Thank you, sir. Thank you. So actually now I think anybody.
Yeah, happy to personally present our product by PPT also.
Yeah.
Give the permission from her.
Yeah.
1 minute. Congratulations. And actually, I would like to suggest Department of Biotechnology has an organization called Biotechnology Industry Research Assistance Council. They are giving you grant if you can combine with any small industry to make it as a formulation and bring it as a product. Easily, you can get up to 50 lakhs.
Okay. Thank you. So, Dr. Mishra, Dr. Sushil Kumar.
Dr. A.S. Rao wanted to say something.
Dr. A.S. Rao sir, boliye sir.
Sir, firstly, I appreciate Kathirasan for a nice presentation. Only one small question is, I also worked on the process control. We successfully failed to control it by physical or chemical means. And my question is, have you observed any biological one, that is natural incidence of pests and disease on processes? To my observation, I have not observed anything. Have you observed anything in your state? Okay.
No biological control.
Okay.
Sir, I have one suggestion.
Yeah.
Sir.
Tell. You tell. Please tell.
Actually,
quickly.
Sir.
Boliye.
Regarding this control of Indian invasive weed, we should also have a massive awareness program, I think, because we can these alien invasive weeds are spreading very fast in new areas. So at least if we are able to, as Dr. Yaduraju was telling, that we should convince the economic losses caused by these weeds. Like even Felis minor, it is just spreading to new area also, sir.
Yeah.
Where wheat was not being grown, and now wheat is being grown, and it is spreading very fast. So if we are able to convince our government, and like on roadsides also, like any big boards we can have, and then we can stop this further spread of these alien invasive weeds, that will be a very good thing, sir.
Yeah, sir. Yeah. So now we have come to the end of this session now.
Yeah.
Dr. Sushil Kumar. Yeah, should I?
You may invite special comment by Dr. Bhagirath,
Dr. Bhagirath Singh Chauhan, I think you can have, if you have.
Dr. L. Rao is also there.
Dr. Rao Saab.
Yeah, no, I don't really have any comments, but I think it was great presentation. Well done, but I just posted there. I thought it's going to finish. So your job is not done. You should really delay your retirement, Dr.
Yeah.
Okay.
And
Yeah.
I think, no, that's good. I think I have got one comment, because now we are talking about all different weed species, these invasive ones. Why don't we have not really a scientific, but we should have a small booklet or somewhere where we actually review what information is available all on different weed species, whether it's allelopathic or biological control on these 10, 12 weed species. And then we were discussing about this importance. And I think that will be a good starting point. And I'm not saying it should be very scientific review, but it will be a good starting point, because I have been working with cropping weeds, and I think now I am expanding. There are students who wants to work on different environmental weeds and rangeland weeds. So if we have some kind of document, I don't know, there may be already, but where we have got information on all different aspects, and where there is a gap also about their impact, as Dr. Yaduraju mentioned, impact in terms of not only just rupees, but also in losses, anything. So I think if we have that information, it will be a good starting point. Yeah, I won't take much time. Thank you very much.
Thank you, Dr. Chauhan. Dr. Chauhan.
Dr. Bhagirath, I want to inform you that long back, about in 2004 or 5, Dr. Yaduraju, sir, he brought out one publication from when he was in DWR as a weeds of, you can say, some invasive weeds, on some invasive about.
We organized a conference, a national conference in Assam Agriculture Institute, Jorhat, and we took people to Kaziranga National Park to show how the whole park has been invaded by mimosa. I'm not Mimosa pudica, but
rubicalis.
Mimosa, some species.
Rubicalis.
Rubicalis or Invisa rubicalis. So I think because of invasion of this weed, the Kaziranga National Park, the rhinoceros, which is an endangered species, is not getting enough fodder. It is a vegetarian, unfortunately. So they're all being driven out of the park, and they're getting killed, by poachers or by accidents on the railway track and things like that. So we wanted to sensitize the game sanctuary people, and we did that in I don't know the exact year, but 2002 or 2003, and we brought out a small booklet. It is not a review one, Dr. Bhagirath Chauhan. It's not a review one, but a small one, very attractive publication on invasive weeds. I think Dr. Mishra may send you a copy if he still has some in his collection. Dr. Mishra can share it with you. And this is more for creating awareness, not for any review type of article. I think, but anyway, good beginning was made, and your point is well taken. I think we can certainly write up review articles on each of these major 4, 5 invasive weeds, and then find out the gaps, and then people can start working on how to take the work forward. Thank you.
Yeah, just last point. In our environments, I have seen, at least in my life, in 20 years' time, weeds are changing their biology. So I think anyway, 2004 to 2021, I think in 20 years, many things have changed. So I think we should really update. And I'm not really talking about review as a paper. I think there's something like from DWR, they have got like booklet, where we have got all that information on those different aspects. I think that will be good.
Yeah.
Thanks.
So what thing, sir, what Dr. Chauhan told, that also I wanted to share, because weeds have changed their biology. Because so far, so many weeds we used to call that annual, like summer annual, winter annual, like rainy season annual. They are not even now that are season bound. They have become now, these are, for example, Transia portulacosta. It was used to be present only in summer and rainy season, but in winter also now it is in Delhi. There are many other weeds. I can tell you there are large number of weeds. They have changed their habit. They have changed their growing habit also. They are coming all through the year, around the year. So that sort of things are happening in the crop ecosystem. Yeah. Dr. Sushil Kumar. So can we extend for 5-10 minutes, 5 minutes or so?
Yeah. In discussion always, that's why you have that liberty.
Yeah. So because time is up to 12:30, so that's why I was worrying. So whether we will, because we have approached that time, not 12:30.
We are having 30 minutes.
Yeah, so
we always end
with,
up with
data
these
we are
senior
having.
people always end up speaking. So I think better to give opportunity to
Yeah.
students and other newcomers.
Yeah, that's I'm telling that.
So
Yeah,
that,
some any
better.
student in chat box all have sent their good wishes to Dr. Kathiresan for his nice presentation. But so far I didn't find any questions or query from the novice scientist or this younger scientist. But so far, if anybody is everybody wants to have any interaction with the speaker, please come up and I invite you all.
Okay, if there is none, then you can go ahead.
Yeah, I don't know. So Dr. Sushil Kumar, so this is the now we have come to the end of this session. So this is now turn of vote of thanks. Should I present? Or you want to say something, you want to speak something? Dr. Mishra.
Okay. Just I want to thank Dr. Kathiresan for a nice and wonderful presentation. And of course, he has stated that he has worked on some species of invasive nature, but still we have to all weed scientists have to work on other invasive importance of the weeds. Still, if you find that in aquatic situations, a part of this water hyacinth, alligator weed is also assuming a serious status in India, as well as this Pistia statuites. This is also one of the very invasive weeds in aquatic situations. And likewise, a part of Parthenia, Mycenia, Chromalena, or Lantana, there are some other weed species are also coming. Particularly, if we talk about the agriculture ecosystem, we will see that this Leptochloa chinensis, for example, he has taken that he has shown that slides. But there is, I think, need to add more alien invasive weeds in that that list. Because so far that list was having only 173 species. But of course, in that Dr. Sandelian, he took all those parameters which qualify for the invasiveness characteristics. But still, I find that there are many species have entered under that criteria, which are alien as well as invasive and has entered into the crop system. So I think in due course, the DWR will take, as suggested by Dr. Yaduraju sir, that DWR will take such those species and try to add in that list.
Yeah.
With this comment, I think you may give a vote of thanks, Dr. Das.
Yeah. With this also, there are so many weeds also that Ageratum hostronianum in Himachal Pradesh, like that. Ageratum hostronianum. Yeah, there are many weeds also there. And certain year they come up in a virulent form. Not that every year, every time they are coming. Certain year based on that climatic interaction.
They have given the
rainfall, soil moisture, temperature interaction. Sometimes they are coming up. Not that they are their presence or their spread is uniform every year. All these things. Sometimes it is happening because of the environmental interaction. Okay, I think with this, I think I come to the. I feel highly, Dr.
Dr. Das asked Dr. Mishra to give his
comments.
Ultimately, he's the man. He's the man who moves things.
Dr. Mishra. Yeah, that's why I was. Yeah.
Thank you so much, sir.
Dr. Mishra sir.
Dr. Das, I also appreciate and congratulate Dr. Kathiresan for a very comprehensive and informative talk on alien and invasive weeds, covering almost all aspects of invasiveness like climate change effect. There are sleeper weeds and weed risk analysis, and more so that the mass awareness, as already pointed out by Dr. Manoj, mass awareness is also important.
Correct.
At the same time, what Dr. Yadraju Sahab has pointed out, that the area coverage of the some 8 to 10 important alien invasive weeds, economic and environmental impacts. Certainly, sir, we want to include these things in our ECRIP program. Actually, as we all know that the Alien University beads, they are the location specific in different, say for example, Mimosa in Assam, like Lantana in some other states. So whatsoever states these Alien University beads are prominent, certainly we are going to include in the technical program of the AICRP bead management, so that at least they can start with the compiling the area coverage, the economic and environmental impact. And the same is the case with the Phalaris minor. For example, it has developed resistant to 7-8 herbicides. So how much the area of the resistant Phalaris minor in Punjab and Haryana. So like that, it will be included in those technical programs. And along with that, we are also preparing for the ad hoc projects, and we will submit it to DST or DBT. So let us see. And for biological control, as you pointed out, we are already in the collaboration with the Bengaluru. But because of the COVID, I think last year they failed to get the new species. But again, Dr. Sushil Kumar is in close contact with them, and we are writing them. And certainly we are hopeful that we can get some more insect for the, especially for the Parthenium. So with this is my comments. And with this, thank you very much.
Thank you, Dr. Mishra Saab. Thank you. Hello, Dr. Kathiresan Saab.
Dr. Das, 1 minute.
Yeah.
There was a question in the chat session whether Chris Parker model can be applied for water hazard. See, for example, all these models are only for assessing weeds potential or a plant's potential to become invasive before it becomes invasive. See, water hyacinth has crossed all borders and all levels, and it has now been established as a very much invasive plant species in the aquatic systems of almost all the states in India. There is no state that is an exception for water hyacinth. So you don't need to apply Chris Parker model for water hyacinth. Chris Parker model can be applied to a new aquatic plant if it is to be introduced into India in the case of an ornamental or in case of accidental introduction or anything like that. Say, for example, one mention was about alligator weed. We can apply Chris Parker model to alligator weed because alligator weed has not become that much invasive like water hyacinth. It has not crossed the third level like establishment. It should expand and it should explode to become invasive. Alligator weed is only in third level, so we can very well apply Chris Parker model to alligator weed. Otherwise, for water hyacinth, it has already exploded and it has already become invasive. No need for applying Chris Parker model to water hyacinth. Thank you.
Yeah,
Thank you, sir.
Yeah, thank you, Dr. Kathiresan. So now I feel highly privileged and honored to propose a vote of thanks. I take this opportunity to profusely thank Dr. Sushil Kumar and Dr. J.S. Mishra for their wholehearted support for organizing this webinar. We also convey our sincere thanks and grateful gratitude to Dr. Kathiresan, learned speaker of this session, and also convey our gratefulness to Dr. N.T. Adaraju, Dr. A.N. Rao, Dr. Chinnuswamy, and whosoever are present over here, that veteran dignitary weed scientists, and their blessings and their suggestions will take care and we'll have to implement in our program also. And I also convey our deep appreciations and sincere thanks to Galaxy of distinguished scientists, colleagues, ladies and gentlemen, and students for their benign presence and participation. And I also thankful to event management host, especially Mr. Gyanendra Singh, for his technical assistance towards successful completion of this session. At last but not least, I must say all cannot be mentioned, but none is forgotten, whose significant contributions made this event successful. And also, once again, I thank one and all. So with this, we came to the end of these sessions, and this was very interactive and useful sessions. And Dr. Kathiresan, I profusely thank you once again.
Thank you.
Thank you very much,
Thank you all.
sir.
Thank you all.
Thank you, sir.
Thank you to all.
Thank you, my dear colleagues, everybody. Thank you very much, Dr. Sushil. Thank you. Bye.
Thank you.
Thank you, sir.
Thank you, sir.